Good afternoon to all. Hope you are all well there. I am Dr. Athul Sandheep, Assistant Professor, Department of Microbiology, St. Mary's College, Thrissur. My topic is on today is steady state, synchronous, and diaoxic cultures. The steady state culture of microorganisms. You have studied in previous class about the four stages of bacterial growth curve: the lag phase, the log phase, stationary phase, and a death or decline phase. The exponential growth lasts only for a few generations and then it leads to a stationary phase. However, it is possible to grow microorganisms in an open system, a system with constant environmental conditions maintained through continual provision of nutrients and removal of waste, which leads to an stationary phase. These conditions are met in laboratory by continuous culture system. A microbial population can be maintained in the exponential growth phase and at a constant biomass concentration for extended periods in a continuous culture system. There are mainly two types of continuous systems has been used in microbiological laboratories. The first one is the chemostat and the second one is turbidostat. A chemostat is a growth vessel into which fresh nutrient is delivered at a constant rate and cells and spent medium overflow at the same rate. Thus, the culture is forced to divide to keep up with the dilution and the system exists in a steady state where inputs match outputs. The chemostat is attractive due to the enormous amount of control that is possible in that. Growth rates, cell density, and selection pressures all are independently set in a chemostat. The culture medium for a chemostat possesses an essential nutrient, example an amino acid, in limiting quantities. Because of the presence of a limiting nutrient, the growth rate is determined by the rate at which new medium is fed into growth chamber and the final cell density depends on the concentration of the limiting nutrients. Other uses of chemostats. Chemostats are also being used for studying aspects of cell biology such as ammonium toxicity. It is used to study the growth rate control, and also chemostat is used for the comparative studies of gene expression for mutant that it is very difficult to compare due to the profound growth rates differences. You can see the structure or function of a chemostat here. The reservoir of a sterile medium, flow rate regulator, air filter, overflow chamber, growth chamber, all you can see this. This is the working process principles of a chemostat. Diagrammatic representation. Chemostat applications: growing large amounts of cells, industrial productions of vaccines, pharmaceuticals, hormones, long-term studies of specific growth phase, selecting for specific mutants, aquatic systems, all these are used. Okay, then we will move on to the second step of a continuous culture method, that is a turbidostat. The second type of continuous culture system is a turbidostat, has a photocell that measures the absorbance or turbidity of the culture in the growth vessel. The flow rate of media through the vessel is automatically regulated to maintain a predetermined turbidity or cell density. The turbidostat differs from the chemostat in several ways. You can see the diagrammatic representation of a turbidostat. Reservoir of the sterile medium, value of controlling flow of medium, outlet for spin medium, light source, photocell, read the turbidity. Okay. The dilution rate in a turbidostat varies rather than remaining constant, and it is culture medium lacks a limiting nutrients, but it is present there in the chemostat. The turbidostat operate best at high dilution rates. The chemostat is most stable and effective at lower dilution rates. Continuous culture systems are very useful because they provide a constant supply of cells in exponential phase and growing at a known rate. They make possible the study of microbial growth at very low nutrient levels, concentrations close to those present in natural environments. These systems are essential for research in many areas. For example, studying interactions between microbial species can be studied through these systems under environment conditions. Continuous systems also are used in food and industrial microbiology. Continuous culture by turbidostats. Okay. Then the major differences between chemostat and turbidostat. The major difference between chemostat and turbidostat is chemostat is a type of continuous culture system in which the flow rate is constant. But in the turbidostat, which flow rate does not remain constant and specific growth rate controls internally by cultural reactions. In chemostat, dilution rate is constant, but turbidostat, the dilution rate varies. These are the major differences between chemostat and turbidostat. Both of these are used for continuous culture methods. The continuous culturing of microorganisms. Now we move on to the second type of bacterial culture, synchronous growth. A synchronous or synchronized culture is a microbial culture or cell culture that are all in the same growth stage. Thus, the entire population is kept uniform with respect to growth and division. But practically, it is not possible to determine a single bacterial cells to obtain the information about growth behavior. Synchronous culture provides the entire cell crop in the same stage. Synchronized culture provides information on measurement made on such culture are equivalent to measure made on individual cells. Now we discuss detail on the synchronous growth. Synchronous cultures are cultures in which most of the organisms are in the same stage of the cell division cycle. This is the one of the important factor of the synchronous growth. All the cells are in the same stage of division. Then various methods of producing the result have been used, but they can be categorized either forced or mechanical selection methods. Forced methods are producing synchrony utilize some artificial means of inducing the organism either to divide or to retard cell division. Double temperature shocks, both not hot and cold and combination of heat and cold have been used to induce synchrony. Another class of forced methods employs nutritional techniques such as following the growth medium to become depleted with respect to one of nutrients and then transferring the organs to fresh complete medium. Addition of thymine to organism which have a nutritional requirement of thymine has also been used to induce synchron. Problem with the forced methods. The important problems of forced with important problem faced in the forced methods is that they interfere the normal metabolism of the cell and data obtained from this type of study must be interrupted with the reservation in mind. Mechanical selection methods depend on some means of selecting organisms of a given size. Transfer of these organisms to fresh medium frequently results in synchronous cell divisions. The most commonly used selection method is filtration through fine filter paper, although differential centrifuge has also been used. The probable reason for success of mechanical method is that cell size changes during the cell cycle and therefore cells of the same size are in the same part of the cell cycle. Synchronous division never lasts for more than 4 or 5 generations and may occur for only one generation. The reason for this is that the generation types of dividing cells vary considerably with respect to the individual cells. You can see a graph of synchronous and asynchronous growth. You can see the diagrammatic representations of an apparatus Elmster Cumming technique for obtaining synchronous culture. Culture filter, invert filter fresh medium, loosely bound cells washed away, new bacterial cells at the same stage of growth and division cycle is obtaining. Then we move on to the dioxic growth. In dioxic growth, growth on two carbon sources. Mixed sugars, each sugar used separately, glucose always used first, second sugar only used when glucose gone. Dioxic cultures. Dioxic growth or dioxy is any cell growth characterized by cellular growth in two phases and can be illustrated with adioxys growth curve. Dioxic growth means double growth is caused by the presence of two sugars on culture growth media, one of which is easier for the target bacterium to metabolize. See, in a dioxic culture media, there are mainly two types of sugars are present. In one of them is first, it is used first, then after depleting, the second one will be used. Dioxy growth is diphasic growth represented by two growth curves inverted by a short lag phase produced by an organism utilizing two different substances or one of which is glucose. For example, we can see when E. coli grows in a medium containing both glucose and lactose, it uses glucose preferably until the glucose is exhausted. Then after a short lag phase, during which bacterial synthesis enzyme needed for lactose use, then it utilizes the lactose. അപ്പോൾ ഒരു മീഡിയത്തിനകത്ത് തന്നെ അതിനകത്ത് തന്നെ കൾച്ചർ മീഡിയക്കകത്ത് രണ്ട് ഷുഗേഴ്സ് ഉണ്ടാകും. ഒന്ന് ഗ്ലൂക്കോസ് ആയിരിക്കും, പിന്നെ വേറെ ഏതെങ്കിലും ഷുഗേഴ്സ് ആയിരിക്കും. ബാക്ടീരിയ ഫസ്റ്റ് ഗ്ലൂക്കോസ് യൂസ് ചെയ്യും, അതിനുള്ള ആവശ്യമുള്ള എൻസൈംസ് ഉണ്ട്, ഗ്ലൂക്കോസ് യൂട്ടിലൈസ് ചെയ്യും. അവസാനം ഈ ഗ്ലൂക്കോസ് തീർന്ന ശേഷം ചെറിയ ലാഗ് ഫേസിന് സമയമെടുക്കും. ആ സമയത്ത് അടുത്ത ഷുഗർ, എക്സാമ്പിൾ ലാക്ടോസ് ആണെങ്കിൽ ലാക്ടോസ് അല്ലെങ്കിൽ വേറെ ഷുഗേഴ്സ്, അത് ഉപയോഗിക്കാനുള്ള എൻസൈം സിന്തസിസ് ചെയ്യും. അതിനുശേഷം അത് ഉപയോഗിക്കാൻ തുടങ്ങും. സച്ച് ഗ്രോത്ത് ഈസ് കോൾഡ് ഡയോക്സിക് ഗ്രോത്ത് ഓർ ഡൈഫേസിക് ഗ്രോത്ത്. Then after a short lag phase during which bacteria synthesize the enzyme needed for the lactose use, growth resumes with the lactose as a carbon source. If the diphasic growth of E. coli is plotted in respect to bacterial density against time, two growth curves follow one after the other interrupted by a short lag phase to produce dioxic or diphasic growth curve. അവിടെ എന്താണ് സംഭവിക്കുന്നത് നമുക്ക് മനസ്സിലാക്കാം. ഫസ്റ്റ് ഗ്ലൂക്കോസ് യൂട്ടിലൈസ് ചെയ്യും, അതിനുശേഷം ഒരു ലാഗ് ഫേസ് വരും. എന്തുകൊണ്ടെന്നാൽ ദ നെക്സ്റ്റ് ഷുഗറായ ലാക്ടോസ്, ഫസ്റ്റ് ഗ്ലൂക്കോസ് ഉപയോഗിച്ചു, അതിനുശേഷം ഗ്ലൂക്കോസ് തീരുമ്പോൾ അവിടെ ഗ്രോത്ത് കുറച്ച് നേരം നിൽക്കും, ഒരു ലാഗ് ഫേസ് ഉണ്ടാവും. എന്തുകൊണ്ടെന്നാൽ അടുത്ത ഷുഗറായ ലാക്ടോസ് ഉപയോഗിക്കുന്നതിന് വേണ്ടിയുള്ള എൻസൈംസ് എല്ലാം ഒന്ന് റെഡിയാക്കണം. പിന്നെ ലാക്ടോസ് ഉപയോഗിച്ച് വീണ്ടും അതുപോലെ ഗ്രോത്ത് കണ്ടിന്യൂസ് ആയിട്ട് തുടങ്ങും. നമുക്ക് ഈ കൊളയുടെ ഇതിനകത്ത് ഇത് കാണാൻ പറ്റും. It has been demonstrated that E. coli growing in a medium containing both glucose and galactose producing dioxphase growth curve is case of glucose and lactose. Similar response has been found in case of other sugars such as arbinose, maltose, sorbitol, etc. when they are used in combination with glucose by E. coli. This sugar is utilized only after glucose has been used in the growth medium. അതാണ് ഡൈഫേസിക് ഗ്രോത്ത് അല്ലെങ്കിൽ ഡയോക്സിക് ഗ്രോത്ത്. അപ്പോൾ ഡയോക്സിക് ഗ്രോത്തിനകത്ത് രണ്ട് ഷുഗേഴ്സ് ഉണ്ടാകും. ഒന്ന് ആദ്യം ഉപയോഗിക്കും, പിന്നീട് അത് തീർന്നു കഴിയുമ്പോൾ രണ്ടാമത്തെ ഉപയോഗിക്കും. അപ്പോൾ നമുക്ക് ഗ്രോത്ത് കർവിനകത്ത് രണ്ട് സ്റ്റേജ് ലാഗ് ഫേസ് കാണാൻ പറ്റും. ഓക്കെ, അപ്പോൾ നമുക്കിവിടെ 3 മെത്തേഡ്സ് ആണുള്ളത്. കണ്ടിന്യൂസ് അല്ലെങ്കിൽ സ്റ്റഡി മെത്തേഡ്സ് ഉണ്ട്. രണ്ടാമത്തെ സിൻക്രോണസ് ആണ്, ഫോഴ്സ്ഡ് ഉപയോഗിച്ചുകൊണ്ടിരുന്നത്. മൂന്നാമത്താണ് ഡയോക്സിക്. ഇത് മൂന്നുമാണ് കണ്ടിന്യൂസ് ആയിട്ട് ബാക്ടീരിയ കൾച്ചേഴ്സ് ഉപയോഗിക്കുന്നത്. Okay. Please go through this, and if you have any doubts, just ask me. Thank you.